I know in this room, I don't have to say that we all have a lot of enthusiasm for central dogma. But here's where I'm going to confess I have a personal favorite part, that's replication. So that makes it my special privilege to introduce today's speaker, CJ Lim, who is an assistant professor in the Department of Biochemistry. Prior to coming here to UW-Madison, he did his PhD in Singapore at the National University, and then a postdoc in a little lab, right? A lab of a Nobel Prize winner, Thomas Cech. We have great hopes that today CJ will share with us some of the answers to the marvelous mystery, which is maintenance of telomeres.
Right. Thank you for having me. Thank you everyone for coming over and listening to me share actually two stories. The one that I did at the end of my postdoctoral training with in Czech lab in Colorado, Boulder, and then another story that we sort of find out this discovery story when I started this lab in Department of Biochemistry about 2 years ago. So the title is kind of interesting, illuminating the dark side of human telomere synthesis. I'm going to explain a little bit later. It's going to be trying to make it as close as possible to genetics, so nothing can go wrong when we talk about DNA replication. So where it all begins, DNA replication actually requires RNA-DNA primer to initiate this elongation, right? This is a very standard leading strand and lagging strand synthesis from a duplex DNA. And these orange primers here are actually made of both chimeric primers, RNA and DNA. And it provides that 3' hydroxyl group for the more processive DNA polymerases like epsilon and delta. They can go for thousands of megabases of replication. But all of them requires a starting point, a starting primer to give the 3' hydroxyl group. And the one that's actually more famous that you learn in your textbook or something very unique is probably what you probably remember these Okazaki fragments, right? It's just because the inconvenience of that you have to make a RNA primer again, and these have these small islands of lagging strand synthesis. So what makes these RNA-DNA chimeric primers? So it's by this very unique enzyme. It's not a DNA polymerase, also not just only a primase, it's a combination of both. That's why it's called polymerase alpha/primase. The catalytic cycle is very simple. I'll just break down how it actually can de novo, build the RNA primer and then extends the RNA primer to DNA subsequently to give it that the DNA 3' hydroxyl group. So this is a very standard DNA template here. Pol-alpha primase actually uses its primase lobe or domains to make this very short RNA primer. It has a 5' triphosphate group, and it's about 7 to 11 nucleotides long. So it has a very undefined or unclarified counting mechanism. So this primase actually will be able to count that it makes about 7 to 11 nucleotides, and then hand over this entire substrate to its DNA polymerase counterpart to extend it with another 20 to 30 nucleotides of DNA. And this really completes the formation of the RNA-DNA chimeric primer, hands over to the more processive DNA polymerases. So you can imagine an enzyme like this, have a primase and a DNA polymerase. It's going to be very, you're going to jump through a lot of conformation changes to go through this complicated process. So what do we know about this architecture of eukaryotic Pol-alpha primase? There's actually 4 subunits. 2 belongs to the polymerase and 2 belong to the primase. So there's prim1, prim2, and Pol-a1 and Pol-a2. So 4 subunits, 2 subunits for each RNA making and DNA making function domains. And we always learn it to be bilobal. As you can see here, what I mean by bilobal, and it's connected by this very flexible linker on the Pol-a1 subunit. So that's the idea, right? You can have this lobe here, which is the primase lobe to make the RNA. Somehow you hand it over to the DNA catalytic center in the Pol-a1 to extend the RNA primer with DNA. So in 2016, a group in Nebraska solved the apo state of the human Pol-alpha primase. And it's quite surprising in the sense that it actually is very compact. We annotated here as a prim1 here. Prim1 contains the RNA catalytic center, And then the Pol-a one that we talk about, this is now in the sort of a navy blue, and it has the DNA catalytic center. And what they saw at the time, this is an X-ray structure, is that the DNA catalytic center is buried in the middle of this compact apostate, right? If you all remember, apostate means it's the enzyme that is not yet engaged to its substrate, no template bound, just the proteins. But what is interesting is that, you know, people have this question about how can Pol-a-primase decide to make RNA first? And this kind of compact structure sort of gives it an insight, right? So because this DNA polymerase catalytic center is buried inside, so first dip comes to this RNA catalytic center on prim 1. It's very nice, you know, hypothesis. But where did we come from? So I'm actually, this is kind of a late introduction, but I work on telomere functions in human. So I'm going to start by jumping across to what are telomeres first, just a brief introduction. So telomeres, as you all may remember, they are just, you know, DNA protein structures at the end of the linear chromosomes. They are made of repetitive DNA sequences. In human is TTAGGG or mammals. What is really unique about telomeres is that it's made up of two kinds of DNA structure. You learn about duplex DNA and you learn about single-stranded DNA. Well, telomere has both. Yes, double-stranded DNA that, you know, spans repetitive sequence of the same kind, about, you know, several kilobases long, and it ends with this 3' single-stranded fill. People often as the G-overhang just simply because the sequence is TTAGGG, majority is G. That's why people call it the G, sort of a G-overhang. This is a description of the structure. It's much shorter. It's several hundred nucleotides long. So why did the cells evolve, you know, to make something so unique, repetitive, double-stranded with a 3' overhang, right? Well, the purpose is that it needs to protect the chromosomes. Our chromosomes needs a protection from false recognition as a broken ends, right? And we'll talk about how telomeres can achieve that. So here's a very standard chromosome metaphase spread, and the telomeres are stained pink color, just so you all know telomeres at the end of the chromosomes. And it really acts like a lack of better analogy is like the shoelaces that you can see here. There are eaglets that protect your shoelaces from being frayed. So telomeres sort of act something like that for the chromosome end. But if you forget about all your molecular biology, telomere actually, you might recall something about telomere from the newspaper. For example, telomeres are often associated with, you know, younger cells or older cells. So if you look at your telomere length when you're a baby, you find that your telomere length is a lot longer. And then as you now go in and check again the telomeres, it has a progressively getting shortened. And this is really due to what we know as the famous problem, the end replication problem for linear chromosomes, right? So after every cell division, you sort of will lose a little bit of your DNA at the end of the chromosomes. So instead of losing precious genetic material, the telomeres are now acting like a sacrificial material that you get trimmed off over time. They're repetitive. It doesn't matter if you trim the first one or the last one. But it has consequences to the biology or the human cellular state, as I'll show later. So now the next question is, you know, if you have something shortening, then you must have something that maintains it. Then this is worked by this very special, unique ribonucleoprotein, which ribonucleoprotein means that it's protein and RNA. This is telomerase. Some of you may recognize this. Telomerase actually uses an RNA template to synthesize the telomere overhang. So telomerase extend the single-stranded telomere G-overhang. It uses this template that keeps adding the repeats onto the 3' overhang of telomeres. So now you think about it, telomerase extends the telomeres, and then you have the end replication problem that gets, you get trimmed a little bit every time you go through cell division. So how does it look like with different cell types? So this is a very nice chart here where the y-axis is the telomere fragment length on average, right, in kilobases, and then this is the number of cell division. You can see different cells has, the way of maintaining their telomeres. That's why telomere maintenance is such a big deal. For example, in a germline cells, your telomerase is always activated. So you preserve the length of the telomere. You're sort of like counterbalancing against cell division. But for somatic cells, the telomere is sort of switched off, right? And then your telomeres will start to like decline slowly over time. That's why we, people correlate telomere length with your, physiological age. And then when it comes to a point where it's so short, it goes what we call the crisis. It starts to tell the cells, okay, it's time to move on to die, and then it kills the cells. But once in a while, you accumulate mutations along the way, and you can somehow reactivate the telomerase, and that's how you can immortalize the cell lines, and why 90% of cancer cells are associated with overexpression or overdrive of telomerase. So what do we know about telomere maintenance, the mechanism of it? So we can really break down telomere maintenance or the making of telomeres into two distinct states, pathways. One is the G-strand synthesis, and another one is the C-strand synthesis. I already introduced what is the G-strand. I'm going to talk a little bit about C-strand next. But before that, G-strand, as I said earlier, is made by telomerase, and telomerase is actually helped by another major telomeric protein complex known as the shelterin. This is not a discussion for the talk today, but shelterin is a protein complex that's involved in protecting the chromosome ends too. But what is the C-strand? So because every time after the telomerase extend the G-overhang, right, the single-stranded DNA, it has to be converted to double-stranded DNA. The way to convert it to double-stranded DNA is by another major telomeric protein complex known as CST. It's a trimeric protein complex that actually will recruit Pol-a-primase to the telomeres, to this newly synthesized G-overhang, and perform what we call the complementary C-strand synthesis this way now. We call it a fill-in process. So there's this G-strand extension and then a C-strand fill-in. So in 2019, I sort of had a question is how this CST is used, or how it does terminate telomerase activity. We should probably jump over a little bit. At this point here, CST is actually able to kick telomerase out. And one way it can do it is by competing with telomerase for the telomere G-overhang. And that's because it has a ability for the CST to bind telomeric single-stranded DNA. So I was looking more on a structure function approach. So if we get a structure, we saw with the CST bound to a telomere overhang, we can understand how CST does its function. And one way we can do is obviously to get a bunch of these purified proteins. So this is a very nice protein denaturing gel of the recombinant human CST that we made. You can see the three subunits, CDC1, STIN1, and TIN1. There's three subunits for CST. In 2019, what do we know about the overall architecture of CST? We do not know the full structure yet, but we know certain domains. First, we know the TIN1 domain is a very simple single OB domain structure over here. And then it has a STIN1, is bind to this STIN1 over here that is this dark blue in color. And then it has a C-terminal domain of STIN1, which is a helix-turn-helix wing domain, two of them. So we saw this, people saw the structure of STIN1 and TIN1, but not so much of CDC1. Well, they only have one structure, a subdomain of the structure at a time, which is the OB domain here. And it doesn't really tell a lot of information of how these three subunits come together to bind single-stranded DNA. And then that's where we have this resolution revolution from Cryo-Electron microscope, which is basically in the structural biology. So we can use this technique called single particle analysis. So once you have all these purified samples, you can actually get a high resolution 3D reconstruction of them if you embed them in this very thin layer of vitrified ice. So this transparent ice, and you can use electron microscope, which shoots the electron beam, and then use a camera to take many images of your individual protein complexes. So you capture them in different orientations, and you can cut out these little boxes, and you can do a lot of, I would call it image wizardry to sort of reconstruct these from a 2D image back to a 3D structure. So that's how people get using Cryo-EM to get a high resolution 3D structure of your protein of interest. But like all structural biology technique, this Cryo-EM single particle analysis is the iterative process, right? You can see it's like a vicious cycle here if somebody's have done before some structural biology work. So like I say earlier, you get to purify your proteins, you have the material, you need to have a certain level of homogeneity, you need to know that it's active before you pursue its structure. So one quick and easy way for you to check the quality of your proteins or how homogeneous your conformation of the protein is by this negative stain EM. I'm not going to talk about how negative stain EM works, but it's a very very cheap and easy way, which I'm going to show a little bit earlier. And if negative EM sort of give you a green signal and say that, okay, you have a really good stable structure here, that's where you go to this Cryo-EM data collection, where you don't have to use any stain. And that's where you can get the highest resolution, the possible resolution for your sample. But the fact is, at this stage, Cryo-EM is actually a point where you can burn a lot of money through that. It's not a very easy and cheap process, so you want to be careful how you proceed it. So I always recommend my students and colleagues that, negative stain EM is always the fastest way. It's just only a couple of dollars. You can actually, see your individual particles here. This is a negative stain EM image of GROW-EL, right? And then how does it look like now in CST, the purified CST that I made? So this is a very a good example of why negative stain EM is a good initial method. So this is a very typical image of a negative stain collection. And then this is where we actually use computer to pick individual particles. The particles are white in color, and this is a zoom in image. You can sort of see like, oh, these particles or this individual protein here sort of look like the same size, right? And somehow the same shape. Why? But then you show it to someone else, somebody will say, no, they don't look the same. Everyone's going to have a different opinion, right? So then we'll leave it to computer. But then to leave it to computer means you give the computer thousands, millions of particles to go and align, right? And if there's a consistent feature or shape, it will come up like this to the right. So that's what we call the 2D class averages from a classification step. You can see that, you can sort of appreciate that now you can see this little man, sort of looking little man-like feature, right? And it's consistent. They all look the same. They're just different class averages, but it seems like they all come from the same thing, which was encouraging at first, and that's why I proceed to Cryo-Electron microscope, do not put the stain anymore, and see how good of a resolution we can get. So the next slide here is really the 2D class averages of a Cryo-EM data set. And you can see again, this looks somewhat similar to the negative stain EM 2D class averages. Just a sanity check to make sure the class averages analysis you get from Cryo-EM has some sort of same shape and dimension as what you got for a negative stain EM. You don't want to have a totally weird thing coming up, right? So what did we get from this kind of data set? So I got a 6.25 angstrom. Nowadays, people like to go out of the 2 decimal place and say they get sub 2 angstrom structure and stuff like that. Anyway, this 6.25 angstrom with a human CST complex. This is actually the first time we're seeing the overall architecture of a CST. As you can see here, this is about 6.25. But there's a problem to this kind of structure. It's kind of nice. We can see it. The problem is that with a 6 angstrom model, and if all the domains of CST has been solved before, then you can easily just dock it in and then you assemble the structure, the atomic model right away. But if we can remember that, we have TAN1, we have STEAN1, but we have a huge portion of CDC1 unresolved. And 6 angstrom doesn't allow me to go in and build an atomic model from scratch. So this is a problem at the time. So like I say, this is a very vicious cycle. I have to go back now to design and think about, how I can proceed with my life at the time. But then I'll always take a trick from, the old school structural biologists is to add in a natural ligand for CST. So what would be a good natural ligand? Obviously, because it binds single-stranded DNA. So I added single-stranded DNA in. I added in 3 repeats of TTAGGG. Initially, when I looked at these 2D class analyses, kind of disappointing. I'm still seeing all these little men. I know this is running to the wall right away. But then I look at this class average here and think this is something weird. At a point, sort of at a time where I either walk out the door, I stay back and reanalyze data, focusing on this large complex here. Luckily, I did that. That's why I'm here. So it turns out that this telomeric single-strand DNA, when I add it in, it triggers a very kind of a dramatic appearance of a super complex. So this is the reconstructed image. This is about 5.2 angstrom. This is really from a very small scale screening data set. But eventually we got to 3 angstrom, and we even found out where CST binds the single-stranded DNA. Remember, I add in 3 repeats. So if you know how to count, 6 times 3 is 18 nucleotides. But I'm only seeing 4 nucleotides on our structure here, which I put it here. But you can see the density beautifully follows the TTAGG model that we made. But at this point, you get a structure, you put a model in 3 angstrom. You show it to someone else, a true structural biologist, they will say, you can put any nucleotides you want in, you will look the same. And then how do you know if this is the real DNA binding site, right? Like, is this the pocket that CST used to bind telomeric single-strand DNA? Well, this is the point where you sort of take advantage of past research. This is where how much you understand your field. Luckily for us, at the time, you just have to map all the human genetic disease mutations that is known to disrupt CST binding to telomere overhangs. And very nicely, all fall near the region of these single-strand DNA binding pocket that we identified. So it's kind of a big relief at the time. We are not, looking at an artifact and stuff like that. But I'm not going to talk too much about this story. Just a really quick summary is that we show that with CST binding to telomeric single-strand DNA, examples, this very beautiful, protein DNA super complex. There's 10 copies of this CST. So we'll go back to the C-strand fill-in mechanism, right? So then you have this, now you have this decamer. We still don't quite yet has the function, right? So why is there a decamer in the first place? Again, people will ask, is this an artifact of Cryo-EM or purified protein? So, but we have this really nice model at the time. We remember that this C-strand fill-in synthesis process is actually a lagging strand synthesis process, right? The Okazaki fragments. You need a lot of Okazaki fragments just because you want to speed up and compare with the leading strand synthesis, which is straight bullet, right? So if the CST is able to form a decamer at a single telomere overhang, then because CST can recruit Pol-a-primase, now per telomere overhang, it can recruit multiple Pol-a-primase, and you can nucleate multiple RNA-DNA primer synthesis to fill in this C-strand process to make it a lot quicker. So that was the hypothesis at the time. So when I started the lab here, that's where I really want to work to do structure function studies of the Telomere C-strand fill-in machinery. This is the model that I talked about earlier, the CST monomer, and then the APO complex of the Pol-a-primase solved in 2016. If we combine these two together, what do we get? This co-complex. So it seems like a very ambitious, 3 subunits and 4 subunits is human protein complex. So I managed to, convince a student on day 1 of my faculty job here that let's go make these co-complex 7 subunits. So very quickly he found that this is not a joke. It's a serious research program. But he actually in 3 months he managed to get this really surprising, and he's really a great student in my lab. He actually made these 7 subunit co-complex. You can see the very nicely resolved 7 bands over here. Of course, when we get this co-complex, the first thing we want to ask is, does it form a stable structure, right? So we go back to negative stain EM, the cheap and easy way of looking if seeing the complex is a stable conformation. So this is the point where, my grad student, he met his first failure. He has the CST, so he did that. This is the APO state of the co-complex that he purified. But what he saw is that from this negative 2D class averages is that you only see either CST or Pol-a-primase alone. And you can compare, this is just a control where we have CST alone or Pol-a-primase alone. We have all these identified out. So there's no new co-complex out. But we know that, he can do pull down, he has verified they can do protein-protein interaction and stuff is like I showed on earlier. But, structural biology is a tough world. While he's doing it, there's this paper that came up from Rockefeller, but they also, but what they saw is, an APO state of this Pol-a-primase CST co-complex. So what they saw is that this is the CST here and the Pol-a-primase here. What they saw is that this APO complex actually doesn't change any of CST or Pol-a-primase conformation. It's basically like taking the structure that we solved in 2020 and 2016 and put it together side by side. Right? So there's not a lot of information given at the time. But what we realized in our lab at the time is, when we throw in a template and let, the enzyme do its de novo RNA primer synthesis, you found that CST actually can stimulate Pol-a-primase, but it cannot stimulate when it's only using 3 repeats. So as we know earlier in the slides that, CST binds to 3x tau really well. But it turns out that if I give it a 3x tau, this is direct assay here, and we titrate in CST, you can see that there's no change in the band pattern or intensity. But if you use a 6 repeat, twice as long template, you see a dramatic increase in the stimulation of the enzyme activity. And over here is just because it's a 6 repeats, there are many ways or many positions that Pol-a primase can initiate the RNA-DNA primer synthesis. They like to do it opposite thymidine. So you can see here, these are the couple of sites that we can assign to all of these products that Pol-a primase made. So now we put in this template, right? The co-complex APO form, we saw no structure. So we just basically reused the same trick 2 years ago. So now there's another surprise. That's why we call it a pre-initiation complex. There's just template bound to the protein. You can see if we put in a 2.5 repeat, 3 repeats, or 4 or 6, or even 3 repeats with a full-back double-stranded DNA, we're seeing this new struct kind of conformation that we never seen before if we are doing, we use CST or Pol-a primase alone. What is really interesting is that, like I mentioned earlier, only the 6x strong enzyme stimulation by CST. Those that is below that, shorter templates, doesn't have stimulation, but still they form these co-complex, which makes us wonder if this is truly a structure that we are trying to solve. Is it part of the enzymatic reaction coordinate pathway? So like I mentioned earlier, this is the pre-initiation complex. It yields a new conformation. Now this is just a side-by-side comparison of CST alone. Now with CST plus Pol-a primase plus the telomere template. And I always like to say this is how it looks like. This looks like a little man here, and then now it looks like a drumstick when I put in the enzyme and the template. So with such a beautiful negative stain EM class averages, naturally it's quite easy to get to the cryo-EM state, and then we solved this about 3.5 angstrom structure of the co-complex, and we solved it. It's not only just one template, but with 3 repeats, 4 repeats, and 6 repeats, and then all of them show the same structure. Essentially, it's 1 CST per Pol-a primase. I'm not going to jump too much detail, but I'm going to go to some of the exciting insights that we got. So what did we see at the time when we get a structure? I remember we saw that structure came up. Immediately it's clear as day how CST can help Pol-a primase stimulate the enzyme activity. So if we just isolate everything, just look at the Pol-a primase alone in an APO state, and then in the pre-initiation complex state that we solved, you can quickly appreciate that CST and template binding by Pol-a primase has split the primase and polymerase lobe apart. Remember last time in the APO state, they're sort of compact, stacked together. Now with this, Pol-a primase engaged with a template-bound CST, it actually segregates these two lobes apart, right? Now you have the polymerase lobe here, and then you have the primase lobe over here. So I'm going to jump into the DNA binding template of CST first before I come back to this story. What we also want to understand at the point is where CST bind telomeric DNA. Remember I showed in 2020 there is only 4 nucleotides out of 18. It's not very informative because CST has a lot of oligosaccharide binding domains, so it should bind more than 4 nucleotides. But so one question that we always like to understand is where is CST binding the rest of the DNA. So in this pre-initiation complex, we actually are able to model another 11 nucleotides now. It's up to 15. You can see this is the original site, 4 out of 18. I said earlier that we modeled in 2020. Now we get all the way up to here, 15 nucleotides. But the resolution for this cryo-EM structure that we got is about 3.5 angstrom, 4 angstrom. So it's debatable how you can, assign the sequence, right? TTAGGG, for example, in 3 repeats, there's so many permutation or register this thing can be assigned to your 3D model. So how can you define that at 3.5 angstrom? So obviously, we have to go back to biochemistry. We basically just do a binding assay across all 6 permutation. We know that this is 15 nucleotides, the minimum site for binding by CST. And you can clearly see that there's only 2 possibilities, the one that ends with TTAGG or TAGG. And then at this point now, just, it's very easy to clarify how these can differentiate just by looking at the cross correlation between the map and the model itself. And we found that the best match is actually by the TTAGG. So of course, people are still not going to be convinced. Reviewers still get angry with how we argue. So we have to go and do metagenesis. Luckily for us, we're able to, use our structure and figure out, the site 2 and site 3 interactions that are essential for DNA binding, and we start to see, some kind of a drop in binding affinity upon introduction of these 2 mutations. So going back to these, the role of single-stranded DNA binding protein, which is a natural cofactor for Pol-a primase. So what is the role, right? It stimulates Pol-a primase activity. So what we actually saw in just a single punchline is that this CST actually sets the stage for Pol-a primase to perform RNA-DNA synthesis. So the first point I'm trying to show just by looking at the structure is that this CST, together with the help of the Pol-a1, is actually able to clamp the DNA template, right? Basically iron out the template, and it really directs the 3' end of the template towards the RNA catalytic center of prim1. So you actually basically clamp it together, and it presents the 3' side of template to the RNA catalytic center. And it's done in a very unique way, because if we remove the DNA template here and just look through from the side now here, you saw that it creates a very narrow and specific molecular tunnel for the single-stranded DNA to track through. And then the cool thing about Cryo-EM is that you can sort of analyze the variability of the conformation that you solve. In this case here, you can see that this Pol-a1 exonuclease domain that's sitting on top of CDC1 is actually sort of like swiveling on top of the template. So we don't know what is the reason yet, but our hypothesis is that it's acting like a Brownian ratchet mechanism that can slide the template forward for the primase to start to de novo build the RNA primer. So I think this point is the most interesting one. We talk about Pol-a primase make this RNA-DNA primer, right? So it has to make the RNA first, then the DNA. So if you look at the arrangement, coordinated by this pre-initiation complex, so this is the 5' end, and then this is the 3' end. This is dashed line is where if we assume that the template extends this way now, you can sort of appreciate that now you have CST is basically presenting the 3' end of the template in front of the RNA catalytic center of prim1. So it allows it to make the RNA primer from 5' end to 3' end. And then now the 3' end is ready to be given to the polymerase DNA catalytic center to extend it with DNA, right? So it's basically sit in the right RNA-DNA synthesis order. So we have this question that we're thinking about, how can Pol-a primase switch from RNA synthesis to DNA synthesis, right? So this is a very complicated process. Previously, people are thinking there's going to be a ton of conformational changes by Pol-a primase. It turns out from our structure, the nature has actually evolved something very elegant. It just uses a single conformation, the one that we solved, to do all of this. Why do I say that? Because we have already known in the past, researchers have shown that this prim2C domain of the large primase domain is important to initiate RNA primer synthesis, because it's able to bind the 5' triphosphate group and the 3' overhang of the template. So I draw in this simplified cartoon. So if you delete, or you remove the prim2C domain of Pol-a primase, it cannot make de novo RNA primer from scratch. It needs this prim2C to be around. But in our structure, we did not see prim2C at all, right? But we know prim2N, we saw the N-terminal half of prim2 is here, and then this is the C-terminal end of this prim2N domain. So presumably that's where the prim2C is going to be. But it's actually positioned in a very nice, poised position between the RNA catalytic center and the DNA catalytic center. So this is like a flexible linker prim2C domain that's able to grab the very first nucleotide of the RNA and the RNA catalytic center here. And then once it's done, it's 7 to 11 nucleotides. You will switch over, you basically hand it over, just a simple swing across for the DNA catalytic center to do its job. So how do we prove this? We just have to go back to our Cryo-EM and then look at our structure again, and we do the variable analysis again. And what we saw is this very flexible loop that is always moving forth and back between the prim1 and the polymerase domain. So this one here, you can see here now in this end-to-end model all superimposed to one another. So this one here is actually the prim2C. So we can do a little bit of 3D classification. Then we can isolate or enrich a subset of our data set that actually can capture the advanced pre-initiation state. So what we saw another structure now here, just a slightly lower resolution, about 4 point something angstrom. But what we saw now is this prim2C docks in front of prim1 RNA catalytic center, as you can see over here now. And this is a side view of how it looks like. We also able to, once we have that structure, we also start to be able to build 5 nucleotides more of a template. Initially we had 15, now we're able to build up to 20. And you can see the last nucleotide that we managed to resolve, will be perfectly poised for if there's an incoming UTP to come in and incorporate. So this all kind of makes sense at this point of time. So if I were to, example all of the information that we have, this is going to be how the template, looks like if it's going to start to initiate RNA synthesis. But what we saw is 20 nucleotides, but in reality, for stimulation, you require more than 20 nucleotides. Remember, I showed earlier is that this is the furthest it goes, the minimum it needs for the incoming NTP to be added. But in reality, you need a little bit more than 20 nucleotides because of the demand of prim2C domain. Remember, I talked about this prim2C domain. It has this is the RNA primer that is making. This was a structure solved in 2016. And then this is the DNA template. So there's this underbelly of prim2C that it needs to engage to stabilize the template. So instead of 20 nucleotides, you need a little bit more. And that's what we saw when we look back at the direct assay now, where we have 3xTel instead of 3xTel and 6xTel, we now put in 4xTel and 5xTel. And we found that 5xTel is the minimum it needs to initiate the stimulation by CST. So this is the portion that I talked about earlier. So then at this point now, we wonder if we can define a template recognition site for Pol-a primase. It's kind of a wild idea at the time because we all sort of learned from textbook, Pol-a primase has no specific recognition site. But now the story will change if it's now partners with single-strand DNA-binding protein like CST. CST has a specific preference for telomeric sequences. So these are our working model now for template recognition site of this vertebrate Pol-a primase when it's coupled with a single-strand DNA binding cofactor. So if you look from the 5' to 3' and what it really needs for the CST version is that it needs the 15 nucleotide of CST binding site that we define in this the work that I talked about earlier. And then there's this spacer. Why I'm saying the spacer here is actually a 5 nucleotide spacer between the polymerase and the primase, where it stretches out to reach the prim1 RNA catalytic center, where it prefers to use the opposite of T or C as the initial start site, the first nucleotide coming on. And like I said earlier, this is not the start site. Beyond the start site, you need an additional 3 to 4 nucleotides for the prim2C to stably bound to hold on the template, and then for prim1 to initiate the RNA synthesis. So I talk about this CST Pol-a primase pre-initiation complex. But then is this, remember this story before about Decama where we have multiple Pol-a primase recruitment? Now the next question is this compatible at all? So this is kind of a messy overlay, but I'm overlaying a diamond of this Decama with the structure that we solved, and it turns out that the primase domain is sort of theoretically okay. It doesn't clash with the thing, but the polymer is going to be a real steric hindrance to the Decama formation. So what could be a possible explanation for this? One is that CST is still able to as Decama recruits multiple Pol-a primase to a single telomere overhang, it can initiate RNA primer synthesis, but once it does that, it sort of have to resolve the Decama and then let the RNA primer continue with DNA elongation. So this is currently a working model in our lab. So a summary for today's talk is really first, I hope everybody will appreciate that this Pol-a primase actually undergoes a huge conformation change when it forms the pre-initiation complex with CST, and this has to be CST bound to telomeric template. What we know is that because CST has the strongest single-strand DNA binding properties, it actually engages most of the template and irons it out. But what is really cool is that CST actually directs the 3' end of the template towards the RNA catalytic center, and that's really how it can facilitate these RNA-DNA primer synthesis order. What is I think it's cool, but we don't have a real function for this, that this Pol-a1 domain, a subunit of Pol-a primase, and CST clamps this template together. This is actually unique. We've never seen this before with other machinery. So we're kind of trying to investigate if this is truly a mechanism where, these two protein complexes come together and translocate the template. It's going to be really cool. But really the punchline is, this structure that we solve really just say that Pol-a-primase doesn't have to jump through a lot of hoops to change conformation, do all these, RNA synthesis, switch and DNA synthesis. You can do all this with just a single conformation. Okay, this is, I think, it's the kind of thing that we looked at it and everything kind of makes sense. So with this end of today's talk, I'd like to acknowledge my group. It's a young group. We are just about 2 years old. This project is led mostly by Qi Xiang and then by Xiu Hua, she's the lab manager. They work really well together, and of course, the entire group basically banding together to come in and help us, help everyone finish up this very nice paper. So I'll be happy to take any questions. Thank you.
I have 3 questions.
Great.
One of which is just a clarification. You could go to, you have this, it looks like an electrophoresis gel plot with like 3x to 6x telomere.
Which this one? Mm-hmm.
So I understand I kind of understand the horizontal plot as increasing concentration of different repeat lengths of the telomere single-strand DNA sequence. Could you run me through the vertical? Like, could you explain?
Right. So, so this is really a de novo primer synthesis gel. So over here is so this is a sequencing gel. So you can literally count, the size of your product 1 nucleotide at a time, and that's what people use for DNA sequencing in the past. So that's why you can assign, 30 nucleotides. Here's the letter, and then this is 32 nucleotides. One of the major bands products made by this Pol-a-primase, right? So what you're seeing here is basically, you have, like you said, different variation of template length, but all of them has a titration of CST, right? So what we really want to see is, at which more precise measurement of at which template length can CST starts to stimulate Pol-a-primase activity. As you can see with the standard 3x tel, you don't see any kind of change, big change, nor the 4x tel. But the 4x tel, if is 24 nucleotides, and based on our structure, it should be enough to stimulate, but it's not, right? Because we say 20 nucleotides is enough to extend, to start to, for CST to help reach into the prim1 RNA catalytic site. But it turns out it's not, because you need additional leader sequence over here, and the minimum you need is 5x tel. I did not show the, the even smaller division between 4x and 5x. But we do have that information precisely saying how much of overhang here, or the leader sequence here that's needed for this stimulation to happen.
So the red on the right is the
assignment of the product size here.
Yeah, so like the RNA primer.
Right.
And what's the minimum RNA primer? Like that's basically what you're figuring out, is that it's like the minimum RNA primer length is like 28, and the other compound.
Well, this is actually a RNA-DNA primer length. So basically, remember, you mix the RNA and then it extends with DNA. So this is the collective length, where it's 32 nucleotides. I think what you're asking is, what is the RNA portion of it?
Or really, what I'm asking is like, what's the cutoff here? What is, when you're saying it's like a sufficient length to activate it or find it, I'm like, what length is that? Right,
the length of the template. So this is the product. So the sufficient length I'm talking about is this blue template here. The products made is based, what you're seeing, these products here is actually the pink and orange. To bind to the template, and then it doesn't need a 3' hydroxyl group. We just start anywhere based on the specificity that we talked about already. It makes the RNA and the DNA. So we traced actually the DNA in our case here. So we are labeling the DNA.
What data cutoff tells you that it's sufficient?
Sufficient.
Like you're saying 4x isn't, but 5x is.
Right. So the cutoff is really just see which template size or length, allows us to see a huge change in, the intensity or the number of products being made. Okay. Does it make sense? Like you can see here, there's no variation, right? Even if I add more CST, but in all changes, when we use a much longer template.
Second question is, if you go back to you were comparing in the like the 5 angstrom section where you had like 16 mirrors, where like you had like a set of 6 alternate possibilities.
Right. I think you're referring to this.
Yeah. So the 2 that bind really well they're off by 1 base. Did you look at the 14 mers and the 16 mers that correspond to the extension and reduction?
Right. That's a good question. We did not try 14 or 16, but past studies have actually shown we don't do it ourselves, but there's been shown that as you drop more, you suffer a loss in DNA binding capability. But then what we really want to chase is that we actually see 15 nucleotides in our Cryo-EM model. The goal really at the time is really try to assign the sequence that we are seeing, right? Whether it ends with a permutation of TTAGG or it ends with GGTT, because you can cycle, you can shift forward, backwards, right? Because we have, for example, in this case, if we use it with 3x tau, it's 18 nucleotides, so you can shift forward, but we also see the same kind of structure when we use 6 repeats. Right, so you can go forward and backward. So now there's only 6 possible permutations. So that's why we test all of them.
Yeah. So an interesting thing about this is you had an earlier result structure pre-complex, which had a TAGG result site.
Right.
And this sequence almost has 3 of those.
Right. I think what you're referring to is this. Site 1, actually, this was tested in that the 4 nucleotide that we saw in 2020. So it's basically this TAGG right here.
Yeah, and then there's almost when you take the 16-mer, there are 3 of those in the whole 16-mer, and those 2 15-mers are the closest to having 3, and none of the others have 3. And I was wondering if that might be important.
You're referring for which structure? This structure or the previous one?
So this DNA sequence has a TAGG and then a GT, TAGG, GT, and then a TAG. But the other 15-mer that binds well shifts it over, and so each of them has 2 of the 4-mers and 1 of the 3-mers that correspond to that motif.
Right. I get what you mean.
3 of them.
Right. I did not present biochemistry, biochemical data on that. That was, I think that was all in the science paper in 2020, but we did test that. But that was a different mechanism altogether, the model for the dimer. Actually, what I didn't show here because, when we found on Dacama, we actually had this idea that the 3x tau 18 nucleotides, even though we only resolve 4 nucleotides, right? But actually, what CST does is the telomeric DNA actually staple 2 dimers inverted together. So use the first repeat and the third repeat of the 3x tau. So I hope that sort of answer what you're trying to ask.
No, I didn't really understand this.
Well, we could discuss more after this. Do you have another question?
So I did have one other. It's a little bit less relevant to the talk, but I was just curious if you had any knowledge or thoughts on the evolutionary origin of telomeres. Because like the evolutionary maintenance is clearly based on the ability to maintain linear chromosomes, but it comes from a non-linear chromosome ancestor, right?
Right. So that's a good question. I'm not someone expert in that kind of thoughts, but I think people talk about transposon, because you can just basically copy and paste, and naturally, if there's an effective strategy to protect the chromosome ends, then this will kind of take off, right, over thousands of years of evolution. Obviously, you need to evolve telomere-specific proteins. There's also another considerations. One thing that we didn't talk about earlier is that when we first saw the structure of CST in 2020, it came to realize that it's actually very similar to replication protein A. In a more specific manner, I think it's almost like a gene duplication of RPA. It's like twice the size of RPA. We still don't know in my lab, we are trying to understand the differences between how RPA engages single-strand DNA versus CST. But that's like altogether another topic. Great. Let me see if there's like three. Anyone has? Yeah.
Yeah, this is a big question. I apologize. When you showed the really nice talk, and when you showed that changes, amazing changes in conformation, like the product fraction, right?
Mm-hmm.
Are you showing us the Cryo-EM and other various forms? Is that how you can see that transition? And is that always a plan, or is there something to actually see that?
Right, that's a good question. So the benefit of this Cryo-EM single particle analysis is that everything starts from a single particle, right? So it's a single state or a conformation. So what Cryo-EM really does is try to align all them, as close as possible to a conformation. But then there's also a variation around it. So what this, variability analysis is trying to put in 3D model, 3D model sense how it actually varies from the most consensus state that we solve. And that's really the power of, single particle analysis. Does that, hopefully that answers the question.
So basically what we're seeing, those transitions are from Cryo-EM.
Right, those are real data, real analysis from our data sets.
And can you model those transitions?
Yeah, I think, I mean, we could, if the resolution is a lot better, we could model. But now we know that both endpoints, I think we could, come in with MD simulation and try to figure out and understand the kinetics involved. But yeah, those are things that we are actually talking with a couple of collaborators on that.
I have a general question. I know it's not maybe related to work, but in general, if telomeres are shortening, where is that extra DNA going? And have you been able to track it with your poly-fragments with the other TFP?
Yes.
Where would that extra DNA end up as a result?
Well, that's a good question. But as far as I know, the end replication problem, you don't really cut the DNA off. It's basically you do not copy end to end. You copy off the end, and then you lose that information that way. That's the classical understanding of end replication problem.
So you can't track.
Yeah, I think that'll be really tough if you want to track, the degraded DNA you're saying in the cells, right?
Has anyone proposed an idea where that extra DNA goes and how it's affected or not?
I don't know. I'm pretty sure somebody's gonna. Right.
So bear with me, I'm a bacteriologist.
No.
I'm wondering, so the original single-stranded plate that's laid down by Telomeres, that must be an error-prone process at some level, right?
Right.
So what happens if there's an error somewhere in here? I mean, I saw you look at the organonucleotide and various, but what if the middle one had a mutation in the single strand? Would that inhibit the switch over from RNA to DNA or the full extension?
Well, that's a good question. I think people has done before, tolerance of CST to a little bit of single base pair change, nucleotide change. There are some key points like we shown earlier, like here. There's some areas that we think, certain Gs, especially Gs, are critical for CST to bind tightly. But this complex actually has evolved, to be able to compensate, a loss in maybe a sequence has some variation. It can still stick. It's like a Velcro. It's not a defined binding site. So it's kind of surprised for us that we can get this much initially because, CST is known to have a ton of single-stranded DNA binding domains. Right, that's why it's so exciting when we get a structure at a time, because every try to figure out where it binds DNA, you chop off one domain, it still binds really well. So something like our replication protein A is a lot of OP domain. You kill one OP domain, another one is going to shift a little bit to compensate. So yes, I'm pretty sure you get a little bit of drop in the binding affinity, but how that change translate to a physiological effect, I think these are good questions and something that people will be interested to know.
I don't see any questions online.
Okay.
Another room with the sexy.
Thank you.